Hello everyone, I am Dr. Pooja, your biochemistry educator, and today we'll be discussing some of the expected questions for your next exam. Okay, so let's start up with the discussion of the questions. Let's see question number 1. Amino acid used in the synthesis of carnitine. Now, this is a very interesting question because in this particular line, we can expect two questions. One is, what is the function of this carnitine? And second is the amino acid which is used in the synthesis of carnitine. So first of all, I'll just tell you, what do you mean by this carnitine? This is derived from lysine. And the function of this is, it is required for the transport of the fatty acid, or you can say acyl coenzyme A, from the cytoplasm to the mitochondria. Because you are all familiar with this, that beta oxidation of fatty acid is confined to mitochondria. And this acyl coenzyme A, because synthesis of fats takes place in the cytoplasm, so it has to be transported from the cytoplasm to the mitochondria. Okay? And carnitine is derived from lysine. So I can say ki there can be two questions from this. One is derivative of carnitine is lysine, and second is carnitine is required for the transport of acyl coenzyme A from the cytosol to the mitochondria for oxidation. Right? Moving on to the second question. So the answer will be A, that is lysine. Then ninhydrin is a reagent used in the detection of. Now see, again, this is a very good and interesting question. Ninhydrin. You know, ninhydrin is actually used for decarboxylation. This is one of the tests which is a confirmatory test for detection of proteins. When we have to estimate proteins, there are two tests. See, estimation is qualitative, it can be quantitative. So here we are talking about quantitative as well as qualitative. So proteins ke liye the confirmatory test, one is biuret, and all of you are familiar with this. Okay, second is ninhydrin. So first of all, biuret and ninhydrin are for proteins, right? And now see, the options are clear. Ketone bodies, blood sugar, blood urea, it comes out to be alpha amino acids. So the answer will be ketone bodies. All of you are familiar with this, that ketone bodies are detected by blood sugar by Benedict's test, right? And blood urea by DAM, diacetyl monoxide test, and alpha amino acid by ninhydrin. There is a complex which is formed, which is known as hydrin dentin. And one more interesting point about this, I'm going to share it with you, that if proline undergoes ninhydrin test, it always gives a yellow color. But except proline, all the amino acids which undergo ninhydrin test, they give a bluish purplish color. Except proline, all will give a purplish color. Right? So this is a very important test for the detection of the proteins or alpha amino acids. Moving on to the third question. Menadione is an analog of. Now see, again, this is a very simple and an important question. Menadione is vitamin K analog. You know, vitamin K has three forms, and this is the only form which is water soluble. And one interesting feature about vitamin K is vitamin K being a fat soluble vitamin, acts as a coenzyme. Except vitamin K, or you can say out of all the fat-soluble vitamins, only vitamin K acts as a coenzyme, right? So the answer will be is K or menadiol. Moving on to the fourth question, total number of dehydrogenases in TCA cycle are. Now see, this is again a very interesting question. Just try to recall what all you have studied in TCA cycle. So I'll just give you the name of the enzymes, then automatically it will be clear. Okay, so actually TCA cycle, some of the students get confused between that link reaction and TCA cycle. So always remember, it always starts from acetyl, always. Acetyl coenzyme A, which is coming from pyruvic acid as a result of glycolysis and link reaction. So this acetyl coenzyme A will combine with oxaloacetic. So remember dehydrogenases, we have to count that in this question. So one is ICD, which is isocitrate dehydrogenase. Then comes alpha ketoglutarate dehydrogenase, succinate dehydrogenase. Right? And the last one is malate dehydrogenase. Now you calculate, first of all, we have to calculate from one molecule. So you can say ICD produces 1 NAD, 1 NAD. This produces 1 FADH2, and this produces 1 NAD. So we can say 3 NAD and 1 FADH2 are produced from one molecule of acetyl coenzyme. Now just calculate. One molecule of NAD on hydrolysis is giving 2.5 TP. So it will be 2.5, 2.5. FADH2 gives 1.5. NAD again 2.5. It comes out to be 2, 3, 4, 2, 3, 4, 5, 6, 7. 5 4s 20, 0, 2, 2, 3, 4, 5, 6, 7, 8. 9. Right? Now you must be confused that in TCA cycle, 10 ATP are generated, isn't it? Now that last ATP, if you talk about, that comes from G. So here they are asking only about dehydrogenases, and there the enzyme is succinate thiokinase. Okay, so we can simply say if they are talking about only dehydrogenases, then it can be isocitrate dehydrogenase, alpha ketoglutarate dehydrogenase, succinate dehydrogenase. Because if you remember, see, oxaloacetic forms acetyl CoA. I'm just writing the abbreviation because all of you know this. Acid, isocitrate, alpha ketoglutarate, succinyl CoA, fumaric acid, malic acid, and oxaloacetic acid. Okay, this is one dehydrogenation. This is second. This is third, and we call it FADH2. So it will be fumaric. Right? And this will be somewhere here when succinyl coenzyme is converted to acid, then comes GTP. Right? So here the answer will be 4 dehydrogenases. And if the question is total number of NADH, so you can say 1, 2, 3. Total number of FADH, 1. Right? This is one cycle. So the answer will be. Moving on to the next question. Eye cell disease is associated with. This is again a very interesting question, and it is a very important question. First of all, cell disease is associated with lysosomes. Basically, what happens is when the function of proteins is over, they undergo degradation. So during the process of degradation, what is happening is they need, whenever we send a letter to somebody, we always put an envelope. Yeah, similarly now with this thing of social media, we are texting a particular person seeing the name, be it WhatsApp, be it Facebook, be it Insta. Same is the case here. Proteins are marked, or you can say they are marked, or the address is mannose 6 phosphate, or it is the address mannose 6 phosphate is the address stamped on a protein to undergo degradation by entering into lysosomes. If that mannose 6 phosphate is absent, obviously what will happen? Proteins will not have their address. They won't be degraded. That is inclusion cell disease. So the answer is A. And the enzyme required is mannose 6 phosphate. Clear? Now, which is the of the following as a coenzyme for acyl coenzyme to malonyl CoA? Now, a very, this is again a very simple question. Acyl coenzyme, malonyl CoA, we are increasing one carbon atom. Whenever we increase a carbon atom, the process is carboxylation. And always remember the coenzyme for carboxylation is biotin. Always. And that is what the question is saying, required as a coenzyme. So the answer would be right. So for carboxylation, it is biotin. For transamination, it is B6. This is again a very important question. See, always remember in biochemistry, always expect, straightforward questions. Clear? Then moving on to the next question, a non-competitive inhibitor. Now see again, non-competitive, no competition, which means this inhibitor can go and bind anywhere. Right? This can bind. It's the wish of the inhibitor. Inhibitor can bind with the enzyme. Inhibitor can bind with the enzyme substrate complex. Whatever happens, wherever it binds, it always decreases Vmax. The Vmax or the velocity will be decreased. But Km will change here. There won't be any change in the Km. So during non-competitive inhibition, normally now you can see C and D option. Students can get confused, decreases or increases. So always remember, you should be sure enough when you are attempting the questions. So always remember, in non-competitive inhibition, Vmax always decreases. Always. And Km is unchanged. There is in competitive inhibition, there is a competition. So obviously Vmax remains the same, but Km increases. Why? Because the affinity of the enzyme with the substrate decreases. Plain and simple. So the answer is clear. Moving on to the next question. Which of the following is an energy requiring step of glycolysis? Now see, glycolysis consists of 3 phases: energy investment phase, splitting phase, and whatever energy is invested, it has to be regained back. Right? So whenever generally kinase is written, it is attachment of phosphate. Right? So if we talk about energy requirement, yeah, you can say energy investment, there is one first step in which glucose is converted to glucose 6 phosphate, and the third step in which fructose 6 phosphate is converted to fructose 1,6 bisphosphate. Here energy is given. So we can say 2 ATP are consumed. And there are 2 other steps in which 4 ATP are gained. That is phosphoglycerate kinase and pyruvate kinase. Now, if you look at the options, these 2 will not be there because they are energy generation. ATP is produced. Energy required is in the first step, which is catalyzed by either hexokinase or by glucokinase. So the answer will be A. Right? Then diagnosis of folic acid deficiency is done by. Now, if we see figure explanation, it's correct. Piglu for aminoglutamic acid. This is one of the intermediate produced during the metabolism of histidine, if you remember. Histidine gets converted to urocanate, and then this figlu, and then this, 4-amino group is given out and glutamic acid is produced. So it is correct. Obviously, peripheral blood smear is also there. And ICAR, this is also one of the very important tests to detect the deficiency of folic acid. So the answer will be all of it. Right? This figlu estimation is a very, very important question. And folic acid, it leads to one carbon metabolism, and it is vitamin B9. One carbon metabolism. Then the last question, glucose transporters in myocytes stimulated by insulin. There are 1 to 12 glucose transporters. But yes, if we talk about mediated by insulin, and that too in the myocytes, it is GLUT4. So GLUT4 is the transporter required in the myocytes. Is it clear? So here I finish up with the discussion of these 10 questions. I hope you liked the way of explaining this. If you like my way of explanation, do subscribe to my channel. And in my next video, again, I will come up with some more new questions related to biochemistry. Thank you so much, guys. Have a good day. Bye-bye.